Hello students, welcome to EPG Pathshala. I am Divas Pradhan, working as a scientist in Dairy Microbiology Division, National Dairy Research Institute, Karnal, Haryana. And today I'm going to talk about genetically modified foods from paper Advances in Food Science and Technology. So we're introducing the topic. Genetically modified foods are foods that has been produced from organisms where specific and desired changes has been introduced into the organism's DNA using the methods of genetic engineering. These techniques are very rapid than the conventionally methods such as selective breeding and mutation breeding, and allows selected individual genes to be transferred from one organism into another, which can be also between non-related species. Genetic modifications allows a greater control over a food's genetic structure that ultimately leads to increase in either quality or quantity of food produced. The technology has also been referred to as modern biotechnology or gene technology, and also it has been popularly known as recombinant DNA technology or genetic engineering. The broad objective of this module is introducing to the concept of omics science. What are the various components of the omics science that interplay in between themselves to bring about the final change, and again the application of the whole concept in nutritional science. Now let us talk about the chronology of GM foods. Advancement in food biotechnology was initially initiated with the discovery of enzymes and their role in fermentation and digestion of foods. Typically, plant and animal extracts were used as a source of industrial enzymes, but were later again gradually substituted by microbial enzymes, which were more on the genetically modified. An example is the use of chymosin in the production of cheese. Cheese was typically made using the enzyme rennet that was extracted from cows' stomach lining. Scientists began using a recombinant chymosin to effect the milk clotting, resulting in cheese curds. Food enzyme production using microbes was the first application of genetically modified organisms in food production. Food biotechnology has grown to include cloning of several plants and animals, as well as further development in genetically modified foods in other areas as well in the recent years. The first genetically modified plant was produced in 1983 using an antibiotic-resistant tobacco plant. In 1994, the transgenic flavor-saver tomato was approved by the United States Federal and Drug Administration for marketing in the United States. The modification allowed the tomato to delay ripening after picking. In the early 1990s, recombinant chymosin was approved for use in several other countries, replacing rennet in cheese making. Till date, most genetic modification of foods have primarily focused on cash crops that is in high demand by the farmers, such as soybean, corn, canola, potato, brinjal, tomato, golden rice, cottonseed oil, etc. These have been engineered for resistance against various pathogens and herbicides and better nutrient profiles. GM livestock have also been experimentally developed, although none are again available on the market. As of 2011, United States of America leads a list of multiple countries in the production of GM crops, and 24 GM crops have actually received regulatory approval to be grown commercially. The basics of gene technology. Genetically engineered plants are generated in a laboratory by altering their genetic structure or makeup in a directed way by either gene addition, that is by cloning external gene, or gene subtraction. In this case, the genes are either removed or inactivated. Plants have been engineered for different characters such as insect resistance, fungal resistance, viral resistance, herbicide resistance, a changed nutritional content, improved taste, and again improved storage capacity. Genetically engineering is a multi-stage process that can be achieved through a variety of methodologies. Once a gene of interest has been identified and isolated from a donor organism, it is manipulated in the laboratory such that it can be inserted effectively into the intended recipient or the host organisms. The manipulation may include changes to the sequence of nucleotides so as to enhance or modulate the expression of the gene once it is introduced into the intended recipient organism. One or more genes of interest, as well as other nucleotide sequences needed for the proper functioning of the gene of interest, may then be built in an orderly sequence into a gene construct. The gene construct typically includes a promoter sequence and a termination sequence, which are actually necessary to ensure that the gene is expressed correctly in the recipient organism. Different promoter sequences control gene expression in different and their own ways. For example, constitutive promoters allows continuous expression of the gene, while others switch expression of the gene on or off at different tissues, organs, and also depending on the developmental stage of the organisms or in reaction to other external influences. A marker gene, that is a antibiotic resistant gene, is often incorporated into the gene construct to help identify which individuals of a recipient organism have been modified by the introduction of the gene construct. Finally, the gene construct may be incorporated into a larger DNA molecule that has been to be used as a vector. Normally, the plasmid or viruses are used as vectors. The purpose of the vector is to assist the transfer of the gene into the host organism. The recipient organism is then transferred or transformed by different methods, which may depend again on our target modification. But normally we have modification using by infection using agrobacterium, particle bombardment, injection, etc., for integration of the gene construct into the genome of recipient organism. The transformed cells are then selected and regenerated into a complete plant. Subsequently, the plants that contains the desired transgene and that express the desired characteristics are selected, which is then followed by crossbreeding to introduce the transgene into other recipient varieties and again multiply it into a number. This figure describes the basic technique of genetic engineering. Genetic engineering starts with the identification of the trait that needs to be introduced into a recipient organism. Once the trait is identified in an organism, it is cut using the molecular scissor that is known as restriction endonucleases. The cut DNA is then inserted into a vector that is normally used plasmids that also includes a selectable antibiotic resistant marker gene. The vector that includes the DNA trait is copied into several numbers in a bacteria, and the vector is then coated into a coat tungsten or gold nanoparticles. The next step includes the loading of the bullet into the Teflon bullet, after which the Teflon bullet is loaded into a gene gun. The gene gun shoots the vector into at very high velocity that penetrates the plant cells. The vector, because of the high velocity, enters the cell, and through different mechanisms, the genes are incorporated into the plant genome. The cells are then plated on a selective antibiotic media, and only cells that have incorporated the vector which is also containing the selectable antibiotic resistant marker, will grow on the selective media. These cells are transferred to medium containing the plant growth factors, and after the full growth, the desired character is expressed in our recipient organism. Now the question arises that why GM foods? GM foods needs a lot of investments from the funding bodies for development of GM foods, typically of the benefits that has been anticipated with the development of GM foods. Some of the anticipated benefits of GM foods are: it leads to creating plants that are better resistant to weeds, pests, and other diseases. It may also lead to an increase in yields of GM foods that can create more efficient use of land, less uses of herbicides and other pesticides. GM foods can lead to foods with better texture, flavor, and nutritional value. GM foods can also give foods that have longer shelf life that can be easily shipped. GM foods can also create an essential sustainable way to feed the world. It has also been hypothesized that GM foods can create agricultural crops that are resistant to adverse climatic conditions, or development of again climate resilient agriculture crops that is very much needed in the growing era of climate change. GM foods can also be used for the development of edible vaccines. The genetic engineering of plants has again seen the potential to provide edible plant vaccines that could be used to immunize individuals against a wide variety of infectious diseases, ranging from cholera to potentially heavy diseases such as AIDS. The health and environmental concerns of GM foods can be allergens. Genetic engineering can unexpectedly transfer pan-allergens and allergens. Pan-allergens are substances commonly found in many plants and have several important biological functions as such in these plants. However, these molecules may be allergic to certain individuals. Some common pan-allergens are profiling, seed storage proteins, and proteases. These are also reports of individuals that are allergic to fish reacting to GE tomatoes that has been genetically altered with flounder fish genetic material. Another point of concern is nutrition. There are evidences that some of the GE foods contain fewer nutrients than the traditional non-GE foods. Since genetic engineering is a technology that can disrupt the tightly controlled network of DNA in the recipient organism, hence chances remain that it may disrupt the nutrient profile of the recipient organism. Another health concern is toxins. Since we know that genetic engineering is not a precise technology and may sometimes insert a new gene inaccurately, now this transfer of genetic material may disrupt the tightly controlled network of DNA in the recipient organism. Now, this process may be able to result in unpredictable effects depending where the new gene will position itself in the host DNA. And this unpredictable effect may transfer very high concentration of plant toxins. Another concern that is well remarked is antibiotic resistance. The use of popularly marker, that is your antibiotic resistance marker in GE foods, is a major concern for alarm. The marker gene for antibiotic resistance is used to determine if they have been successful in inserting the gene of interest. However, that may result again in dangerous levels of antibiotic resistance in humans, since it has been seen that DNA can also transfer from GE foods to the microbes in the gut system of the recipient. Continuing with the risk of GM foods can also lead to immune suppression and cancer risks. GM foods are typically engineered to withstand unlimited application of chemicals, including pesticides. These GM foods hence may carry pesticide residues such as bromoxynil and glycophosphate that have been actually associated with the developmental disorders in fetuses, tumors, carcinomas, and non-Hodgkin's lymphoma. Another risk of GM foods is soil contamination. Since the genetically engineered plants are engineered to resist insecticides, hence insects may take up the resistant gene and become resistant to insecticides, thereby increasing the amount of pesticides used to inhibit those insects. Now, these pesticides, which are generally toxic in nature, may accumulate and build up and lead to soil and water contamination. One of the examples that has been cited before is Bt cotton was genetically modified to produce the Bt toxin and kill insects. Now, studies have found that Bt toxin after 234 days was still present in the soil. Creation of superweeds and superbugs. Using GM foods may also lead to transfer of insecticide resistant genes from GE crops to weeds that again may occur by cross-pollination. This may lead to the creation of superweeds and superbugs, which can be highly resistant to the normal level of weedicide and pesticides, and hence that may ultimately lead to the farmers using more and more pesticide and leading to the other problems that has been mentioned before. Addressing the world hunger, there is a big question about genetically modified foods alleviating world hunger. However, the main reason for the world hunger may be not actually the GM foods, but the problem that is involved with poverty, inequality, and the food excess. The safety assessment process. The safety assessment process starts with a GM food can only be sold legally if it has been assessed and found to be safe and also approved by relevant agencies. The producer of GM food first must apply to a relevant agency for the food to be approved at the commodity level, that is, as a new primary product. And until the food from a particular commodity has been approved, products containing food or ingredients derived from that commodity cannot be sold lawfully. Safety assessments for GM foods should be based on the best current scientific knowledge available and should be carried out on a case-by-case basis. It should also fully consider the safety of each new component in a GM food, and again finally consider both the intended effects of the genetic modification. The first is based on the best current scientific knowledge. The scientific assessment for a GM food should be based on the best current scientific knowledge to ensure that the best possible assessment is made for the food. And to obtain this scientific knowledge that is available, or the literature that we may be talking about, information from a variety of sources that are published scientific literature, general technical information, independent scientific consultation, other regulatory agencies, international bodies, and also the community should be thoroughly investigated. It should be carried out on a case-by-case basis. Again, that is because the safety concerns depend on the type of food and the nature of genetic modification that can again vary depending on different case. GM food obtained from a particular type of GM organism, whether it be plant or animal, requires a separate safety assessment. GM foods cannot be assessed as a single class because the safety concerns will again vary depending on the type of food and again the type of modification and type of genes that has been included. Now continuing with the safety assessment of GM foods. Safety assessment of GM foods needs to fully consider the safety of each and every new component in a GM food that has been introduced by the technology, whether it be a DNA material or proteins. GM foods generally contain both, that is the new genetic material, that is the new DNA, and the new proteins that has been coded by the new DNA. They are new in the sense that they have not been previously existent in the food being considered. Hence, the safety should be considered of each component separately in the GM foods and fully. Another aspect of assessment of GM foods are it should consider both the intended effects of the genetic modification and again the unintended effects. The intended modification may be the presence of new protein, and again the intended effect may be the changes that it brings to the levels of toxins or allergens. The intended effects of genetic modification are those that happen as a direct result of the presence of new genetic material and proteins. However, genetic modification may also have other unintended effects such that it may change the level of toxins, allergenic components, nutrients, or anti-nutrients in a food. The safety assessment typically looks specifically at these characteristics to ensure that there is no major differences between the GM food, the newly developed one, and the conventional equivalent food. Another potential unintended effect may be whether the new genetic material in the GM food could affect the human health by moving from the GM food to the bacterial cells in the human gut. A famous case study of the side effects of GM food is the monarch butterfly effect, the insect that was resistant to the Bt cotton. Decades ago, farmers discovered that a soil bacterium called the Bacillus thuringiensis or the Bt infects and kills the caterpillars that eat their crops. The bacterium produces a protein that is harmless until it turns toxic in the caterpillar's stomach. There, an enzyme cuts the Bt protein into pieces that lock into a special receptor called the protein lock in the caterpillar gut. This locking action destroys the gut and kills the caterpillar. Adult butterflies and other insects and animals don't have any locks for the Bt toxin. Bt doesn't harm wildlife the way traditional pesticides sprays do. In fact, organic farmers have relied on this natural biological pesticide for years. Now, the scientists utilized this and inserted the gene for the Bt protein into crops that are frequently destroyed by the caterpillars. The plants started producing the Bt protein in their leaves. When the caterpillars started feeding on the leaves, they gradually started to die without even the pesticide sprays. Now the monarch butterflies, on the other hand, begin their life cycle as caterpillars, as the larvae. The adults drink only the nectar of milkweeds and they lay their eggs on the milkweed's leaves. Monarch butterfly migrate far and wide throughout the United States, but all return to one small, unique area in Mexico for winter. Now, recently it was observed that the part of that habitat has been destroyed, that has been mainly attributed to the Bt toxin, and additionally pesticide sprays throughout the migration path have further reduced the monarch number. Naturally, people were a bit concerned about the new threats to the butterflies' well-being and as such to the population as a whole. GM foods in the future. In the past, development of GM foods have been focused mainly on the development of cash crops that has been already mentioned before. However, in the future, the GM food crops developed has been discussed in the coming paragraph. Development of new GM crops requires many years of research and development in the laboratory and in the field to discover and understand new traits. At present, only a small number of food plants have been genetically modified, and most of the modification have been for the benefit of growers. For example, herbicide tolerance and resistance to pests. In the future, a wide range of fruit, vegetable, and cereal crops are likely to be genetically altered in various ways, some or majority of which could be of benefit to consumers. For example, modifications could be used to create GM foods with increased vitamin and mineral content or healthier fatty acid profiles. Other traits that could provide environmental benefits, such as crops that are more tolerant to drought, may also be developed in the future. GM animals. Genetic engineering technology can also be used to modify animals as well as plants. Genetically modified animals, that is often also referred to as transgenic animals, that are actually modified genetically in some way or the other. Such animals might have been modified to contain one or more new genes from other organisms. Alternatively, one of their own genes could also have been altered, perhaps by being duplicated or also by getting it removed. The types of animals that are being genetically modified include cows, sheep, goats, pigs, fish, and insects. Animals are being modified to develop normally for pharmaceutical and health related products as well as food and agricultural products. For example, GM foods have been developed that contain a gene for an antifreeze protein, allowing the fish that has been modified to survive in very cold water conditions. The gene for the antifreeze protein was originally taken from another species of fish that thrives in freezing water. The regulatory system for GM animals and their products have been the subject of increasing recent attention and discussion among the media, policymakers, research scientists, and as such the general public. Many countries are now considering how food derived from GM animals might be regulated and what type of scientific data might be required to assess its safety. International bodies, including the FAO, the Food and Agriculture Organization, the WHO, that is the World Health Organization, and Codex Alimentarius, have set up committees of experts to consider this issue. However, as of June 2005, no GM animals have been approved for food use anywhere in the world. Coming to the conclusion part, dear students, today we learned about the basic concept of genetically modified foods and also the technique that is used for the development of various GM foods. Just like all the technique have its own advantages and disadvantages. Similarly, GM foods also have both positive as well as negative points. Hence, it should be kept in mind that if the benefits of GM technology is to be harnessed and taken proper advantage of, then extensive safety trials of the genetic modified foods that has been already mentioned before needs to be properly undertaken before it is released to the general population for the general consumption. Thank you.